Well, hello, everybody, and thank you for joining us for our session. I'm Corey Painter, and I'm the Deputy Director of the Count Me In initiative out of the Broad Institute. It's my distinct honor to host this fireside chat with Dr. Ned Sharpless, the 15th Director of the National Cancer Institute since 2017, and Dr. David Begenbaum, an Assistant Professor of Medicine at the University of Pennsylvania, founding Director of the Center for Cytokine Storm Treatment and Laboratory, Associate Director of Patient Impact for Penn Orphan Disease Center, and last but certainly not least, co-founder and president of the Castleman Disease Collaborative Network. Thank you both for what I'm sure will be an inspiring conversation. In today's chat, we're going to keep with the theme of the day, and we're going to dig into topics tied to data sharing with the goal of surfacing actions that can be taken to better enable policy and practices to drive impact from patient data. The two of you are uniquely suited to offer perspectives that span academia, advocacy, and government. And it's my hope that our conversation will enable all of the attendees to explore what roles they can play to ensure we are moving with a sense of urgency toward more data, better data, and shared data. To start the conversation, I'm going to take a couple minutes and I'm going to talk about a patient-partnered genomics initiative that I helped build at the Broad Institute called Count Me In. The goal of this initiative is to generate massive publicly available databases of clinical, genomic, molecular, and patient-reported data in cancer to enable researchers to find patterns in that data, to help accelerate discoveries and the development of new treatment strategies, and to build all of this alongside the very patients that it's meant to serve. My role within this initiative is deeply personal. I'm a scientist by training, but I'm also a cancer patient by terrible luck. When I was diagnosed with an exceedingly rare and aggressive cancer called angiosarcoma 11 years ago, I immediately understood the lack of infrastructure that would be required to make even the slightest dent for those of us who were slammed by a rare cancer diagnosis. What I didn't realize or appreciate at that time was that many of the barriers that I was facing could be addressed if you were creative. They could be addressed by building infrastructure for common cancers that impacted much larger populations of patients. And if built correctly, the same infrastructure could be leveraged for those of us on the margins of research. If we could do this and do it well, we have a shot at democratizing the ability for everybody to participate in research and drive discoveries, and to do it with unprecedented speed. One of the silver linings of my cancer diagnosis, if there is such a thing, was that it absolutely stripped me completely of every single career ambition. I know that doesn't sound like a silver lining, but I promise it was. It gave me unwavering focus on one thing: impact, and impact alone. So I came to Count Me In to build this, which enabled me to focus exclusively on just that. In 2015, I joined the Broad to help build the Metastatic Breast Cancer Project alongside dozens of metastatic breast cancer patients. Together, we built a platform that could leverage the will of the patient to contribute to research wherever they lived through remote consent. This enabled us to obtain medical records, biological samples, survey data from patients that were treated at over 1,700 institutions across the country. We have since generated and released clinically annotated genomic data into the public domain. And from the Metastatic Breast Cancer Project alone, we have over 30 peer-reviewed papers citing that data. We took this platform and pointed it to angiosarcoma, just like I had hoped we would be able to do. And within 3 years, we made discoveries that we published in Nature Medicine, and our data was used in several clinical trials that have since validated our findings and created new treatment modalities that have been practice-changing for many sarcoma clinicians. Patients have incredible capacity to help research efforts, and they're clamoring to drive impact. I just want to plant that seed with every single person listening to this. Patient involvement is an area ripe for disruption, and each of us can play a role by thinking creatively in order to maximize their contributions. This can be through their voice driving policy. It can be through tangible measures, such as acquiring and sharing their samples and data. And it can be through participating in efforts like ours to drive the ability to generate massive amounts of data to be shared. I'm sure there are many additional ways that this whole area can be championed. And in fact, I'd like my first question to touch on just this: the roles that patients and advocates can play to drive research in their own disease. Dr. Feigenbaum, you have an incredible story that resonates so deeply. Would you please take a moment and tell us how you drove generation of data in your own rare disease as a patient, scientist, and doctor? And in particular, how do you think that this was made possible? And what were the biggest barriers?
Sure. Well, thank you so much, Corey. It's such an honor to be on this panel with you, Dr. Painter, and also you, Dr. Sharpless, and to be a part of what an incredible afternoon. I've been so impressed from the very beginning with Dr. Kayla's presentation all the way through, and really just an honor to be a part of this. So just as you mentioned, I have a personal journey like you do with my condition. I was diagnosed with idiopathic multicentric Castleman disease while I was a medical student at the University of Pennsylvania, and I hoped to become an oncologist to treat cancer patients. When I became a critically ill patient myself, I spent months hospitalized. I even had my last rites read to me, and eventually started getting chemotherapy that would save my life. But unfortunately, I would have a number of relapses and really ran out of options. There were no more drugs left. And that's when I went on this journey to create the Castleman Disease Collaborative Network to bring together physicians, researchers, and patients to advance research for Castleman disease, eventually identifying a drug that had never been used before for Castleman's that I began testing on myself. And thankfully, it's been over 7 years that I've been in remission on this treatment. And when I say testing the drug on myself and I say I started this foundation, as you know all too well, it really was an incredible collaborative effort that the CDCN facilitated to bring together physicians, researchers, and patients to make these discoveries. But as you also know, it wasn't easy. There was a lot of knocking on biobank doors. There was a lot of trying to figure out solutions, working with the pharmaceutical industry to figure out what samples are maybe waiting to be unlocked from freezers. But it was through collaboration. It was through recognizing that the system really isn't positioned for collaboration. It's not positioned well for sharing of data and samples, and that we would need to do some radical things, following in your footsteps at Count Me In, going directly to patients to consent them to give us samples and also their medical records, to partner with the pharmaceutical industry. Actually, Janssen Pharmaceuticals has come up a number of times this afternoon. They made biospecimens available from their large randomized controlled trial in Castleman disease. They made it available to our research team, and we just published a week ago a novel predictive biomarker of response to their IL-6 blocker, and even a new therapeutic modality based on this proteomics data. These are samples that would have stayed in freezers and kind of frozen away that we were able to unlock. And so I think it's this sort of collaboration that really has enabled me to be here today and to be on this drug that's keeping me alive.
That's really incredible. Thank you for sharing all of that. Dr. Sharpless, it's clear to me and everybody who knows you that your commitment to data sharing is unparalleled. Dr. Fagenbaum and myself have both experienced firsthand the ability to accelerate discoveries through data generation as a result of a complete realignment of our incentives. I'd love to hear your view on the roles that the NCI can play in cementing the need to share data through changes in incentive structures.
Yeah, thank you, Corey. And I appreciate the opportunity to be here today. This is a topic I really care deeply about. And it's maybe the way I got to this is it just feels sort of frustrating, right? You as a physician, you take care of these patients. You think this guy with leukemia, that can't be that rare. Why can't I just find out what's happened to the other people like that? And then when you come to the NCI and you get interested in childhood cancer, that problem is essentially true for all of childhood cancers. Is why can't we just know what happened to the last 100 kids and have samples and share their data and we'll know. And many of you, I think, will remember Vice President Biden, when he was talking about the cancer moonshot, spoke explicitly on this topic at great length about how this was something that drove physicians, patients, advocates, loved ones, caregivers, everybody crazy, that there wasn't better data sharing, and we should just focus on that effort. And so when I came to the National Cancer Institute, I started asking these questions about how do we share data and what data do we hold. And to be fair, the National Cancer Institute has some very, very successful big data initiatives. The Cancer Genome Atlas would be the prototypical example, for example, which has been used widely for thousands of publications for purposes that nobody ever envisioned when they were, starting out that effort. And it's been very, very successful. And I think it shows that when you make great data available to the research community, it will be used for really great purposes. But one thing I learned along the way is that this is much harder than I thought it was. David touched on the issues like just figuring out the inventories is not simple. People have stuff stuffed in their freezers. Nobody even knows what's in that box until they open it up, right? but there's also these issues of privacy and consent. A sample that may be acquired through a consent that was intended for one purpose, and now we want to reuse it for whole genome sequencing years later. And how does that work? Did the patient really agree to that? And then how do you distribute that data if it was obtained through no consent or through some other very limited consent? So these privacy and consent issues are a problem. And then I think also one thing that isn't really well appreciated is a lot of the quality of the data is not very good. Is that even if we have the data and we sort of know something about it, and then we want to put it somewhere and aggregate it in a way that's de-identified and protected, it's not easy to do that because often the data are like somebody's Excel spreadsheet with field names they made up, and it's just data aggregation. Somebody's got to go through that by hand and manually curate those data. So it's a much more expensive and complex endeavor than I think I realized until I tried to make it happen. So those are the challenges. I think they're well aware of this. This group is well aware of these challenges. What can we do about it? And that's what I think we should talk about today. But I will say one of the things we're very excited about the National Cancer Institute is the Childhood Data Initiative, which is really, I don't think we've ever had anything that's $50 million a year times 10 years, $500 million. That's really so focused on this issue because, as I said, this problem is just throughout childhood cancer research, and having great data and making it available, we think would be very, very useful with proper clinical annotation and genomic linkage, et cetera.
Thank you. You highlight a lot of the different barriers that we have to come together to solve. Dr. Feigenbaum, maybe you could expand from what Dr. Sharpless just said and describe what roles you think advocates and patients may play in terms of figuring out what data even exists so that we can find out how to benchmark gold standards and aggregate and surface what needs to be shared in the first place.
Yeah, I think patient advocates really are uniquely positioned to serve as this sort of quarterback within the medical system. Of course, researchers are constrained in the work that they do. Physicians obviously are constrained. The federal government, there are all these constraints. What academic institutions can do, there are kind of almost these lanes, right, that many of us are going down. And I think patient advocates have the ability to kind of cross between lanes and to basically say, to serve as an intermediate between a pharmaceutical company that may have some data or some samples or some insights and a researcher who maybe, like Dr. Sharpless mentioned, may just have a spreadsheet on his or her computer, and to be able to be that sort of unbiased driver who is at the end of the day so motivated for progress. I mean, you and I, Corey, we've connected over the years, and just the fact that our skin is in the game, and frankly, we have a lot of friends, of course, who have skin in the game with our particular diseases, and that will put us in a position where we're an unbiased advocate, but we also are relentless, right? And so, when we become aware that there's something in one place or another, and the speakers before us were so wonderful at highlighting the solutions to some of these big problems. I think that the solutions exist, as has been mentioned, and maybe they've even been around for a while, but they're not easy. They're expensive, they're hard, and I think that the patient advocate and patient advocacy groups are really well positioned to be that sort of dogged, bulldozer in the fight, to just kind of keep pushing, but recognizing that it's not going to always be easy. And just as Dr. Sharpless said, you may do all of the work, and at the end of the day, you get data that doesn't generate any insights that you can't do anything with. But I think that many of us have really great experiences where we've fought hard to get the data, get the samples, and we have gained something from it.
Mm-hmm. Thank you. Dr. Sharpless, building from that, I'd love your perspective on what we could do if we had more broad consent, if more patients were able to participate in research, and we could then put that knowledge into the hands of advocates to really drive awareness so that more people could participate in research. What are the barriers that you see right now with respect to patients' consent, whether it's the form of the consent or getting it into the hands of the right patients?
Well, I have several things to say on that topic. Maybe the first one to say is that consent is nice. It's good. We all like consent, but you have to be able to do research in the absence of consent, because there are some people who, for a variety of reasons, are not consented for clinical trials. They're not consented for sample collection. They're anonymous to our clinical trials infrastructure. And so we have to learn about what happens to these individuals in the real world. So the requirement for consent and the requirement for participation in a clinical trial is in a way not as inclusive as I think we would like our research efforts to be. So we have to solve that problem. And that is a conversation I think we as a society have to have. I mean, this is I don't think the NCI director should say where our line should be on where we can waive consent, but IRBs and advocates and patients who care about cancer progress have to realize there's a fundamental tension between privacy and research. And privacy is good, research is good, and somewhere as a society we have to draw a line on where we are going to, what sort of research we are going to allow in the absence of consent. But I think we have to be able to do that, or we won't be able to make progress for all of our patients, as opposed to just the ones that want to participate on clinical trials. Then, when a patient is able to give consent, that's a better problem. Although you're not out of the woods yet, because consents are often, particularly in a prior era of research, were quite limited. They were sort of you'll be on this lung cancer trial, and we'll give you this drug, and you consent to that trial for that. And then somebody that later on has samples and they want to, sequence their whole germline genome and put that on the internet. Did you consent for that? So we have to really look at consent and understand what we've asked individuals to do, and is that okay? And would a reasonable individual have agreed to this analysis? Mm-hmm. And many people, then when I explain this problem, say, well, why don't you just call them back up and reconsent them? Sounds like a great idea. It doesn't work nearly as well as one might imagine. It's very difficult. Often we can't track people down. It's expensive. People don't generally like to get phone calls when they've been cured of cancer years ago saying, by the way, can we, sequence your tumor? So it's not always a well-received conversation, suffice it to say. So, consent is really important, and I think makes a lot of this simpler, mm-hmm. but it doesn't solve all the problems, because, this research is sensitive. When you talk about intrinsically identifying information, things like, elaborate electronic health records, as well as genomic information that is private and unique to an individual. Doing research with that has some risk to the individual, and in terms of compromising their anonymity. I think the NCI has gotten really good at thinking through these issues. We have privacy protecting technologies, hashing and matching, and synthetic data and stuff like that. We have data enclaves where we create super secure data structures where one can only get in and compute on the data in the cloud, for example, and not download the data. So we have measures to address these things. Mm-hmm. And I think I believe that we can provide the opportunity to do great research that really addresses the questions we need to address, while simultaneously protecting the individuals who are participating in science. But this is where we need advocacy to weigh in, because there are some judgment calls where we need society to understand what we're doing and what we intend to do.
Thank you for sharing that. I'd love your perspective, Dr. Feigenbaum, on whether or not you ran into any of those issues with consent, and if your life would have been made easier if you were able to conduct any type of research in ways that would have been made, I don't want to say without consent necessarily, but that were not constrained by the current consents of the samples that you were trying to get your hands on.
Yeah, it's a great question. And at the beginning, I alluded to this particular study where we utilized nearly 100 samples from patients that were part of a clinical trial, samples that actually those patients consented for 10 years of research use. And we were closing in on that 10-year period. So literally, they were going to have to be discarded. And that was an example where it wasn't that the patient said, I only want you to do research for 10 years. Just someone had written a consent that said, you can only do research for 10 years on it. So it wasn't necessarily the patients didn't want us to do it anymore, but that's just what they signed off on. And so we got our hands on them around year 8. We proteomically profiled the samples, and we learned a lot. We learned this predictive biomarker signature. We identified a new therapeutic target based on it. But I think it's a good example where we went through all these options. Can we recontact the patients like Dr. Sharpless mentioned? And we learned that many of those patients are no longer alive. In fact, I think half of those patients have died from their disease. So truly, this is their last true legacy on Earth, right? These are samples they gave. The drug may have helped them for some period of time, but now they have an opportunity, or there's a legacy opportunity for their sample to help a future patient. And so we felt really, really compelled to do everything we could to do as much research as we could before that 10-year window ran out. And, one possibility Dr. Sharpless mentioned is that in some societies, you could go to ethics committees or IRBs and say, you know what, there's a 10-year limit here, but we're looking for an exemption here. And it's possible that that could have been given, but we decided to try to get as much work done in that 10-year window as possible. And then the remaining samples actually were discarded. And you know, for someone like me, that's really, really tough as a researcher and also a patient. The idea that these clinically annotated clinical trial samples, high grade samples, truly cannot be used, you know, beyond that period is really difficult. But as you know, we've really gone the route of saying, let's go directly to patients. Let's consent patients directly. In fact, earlier today, I was on the phone with a patient on the other side of the country who's sick in the hospital with Castleman disease, and we're able to consent that patient to our study. We can send tubes in the mail, overnight tubes to the family. The family will bring the tubes into the hospital. Blood will get drawn during clinical draw. These tubes will get added to it, send the tubes back to us. This bypasses the challenges of having to get an IRB set up at every institution in the country, exactly, or having to work with every academic center for transfer samples. And so these sort of direct to patient opportunities, at the end of the day, the patient wants to be a part of this. And this is a great way to work with them in avoiding some of the bureaucratic issues that we've seen.
Yeah, we've had very similar experiences with Count Me In. It's the will of the patient. Nobody wants us to succeed more. Nobody wants us to make discoveries faster. They're definitely the biggest stakeholders. And I think it's incumbent upon us to find creative ways, just like you did. And, you know, I really hope that everybody in attendance is thinking, how could I drive this faster? You know, working directly with patients. What kind of protocol can I write that has, you know, if we are going to, you know, generate data, maximal capacity to share. And I think that we've, you know, hopefully surfaced a lot of issues that can transcend what we're doing here today and get into the right hands, advocates and patients, so that they even understand your tissue and your tumors and your data could be destroyed within a time frame. You know, maybe people, if they're aware of this, they will take action to preserve and drive the research that we need so desperately, so that we can generate the data to be shared for everybody like you. And unfortunately, we have a really quick session today and we are out of time. So I would very much like to thank you, Dr. Ned Sharpless and Dr. David Bakenbaum, and all of the attendees joining us today. Up next is the session, Patient Power, the untapped resource for accelerating discovery, being led by Sue Sherman from the Lamb Foundation. Thank you both so much.
Thank you for the opportunity to speak today.
Thank you so much.